Firstly, I would like to thank the organizers for giving me this wonderful opportunity to speak today. My name is Thandeka Nkosi, and I'm a master's student at the University of KwaZulu-Natal in Durban, South Africa. So KwaZulu-Natal is located in the East Coast region of the country, and we are based in the city of Durban. And here, I'm also showing you a beautiful city of Durban with its beautiful weather throughout the year. Unfortunately, surrounding this beautiful city are mostly rural areas and impoverished areas that have been shown to have high HIV prevalence. So a study conducted by Abdul Karim in 2014 showed that there was a high prevalence of HIV in women who were undergoing antenatal clinic in rural South Africa. For example, it was shown that between the ages of 21 to 22, the prevalence of HIV was 39.4%. So such high prevalence led to the design of the study, FRESH, for females rising through education, support, and health, which is a cohort of young HIV-negative women who are at high risk of acquiring HIV. So the objectives of this cohort was to provide weekly empowerment to these young women, the life skills, job readiness, HIV prevention curriculum, with an aim of preventing HIV infection and allowing these women to be placed in employment, because most of these areas have high unemployment rate. So during their visit, these women will be tested twice a week for HIV RNA by finger prick, with an aim of allowing to identify the hyperacute infection. And as soon as they are identified HIV positive, they are placed on immediate therapy. So far, the study has identified a total number of 56 acute infections, with 14 being untreated and 42 being early treated. So most of these acute infections were identified in FEBIC stage 1. In this figure here, I am showing a typical example of an early treated individual. We can see that when these women are placed on immediate therapy, viremia is suppressed to undetectable level, and CD4 count is maintained over time. However, in a case of untreated patient, we find a viremia that persists over time. Also, the benefit of initiating antiretroviral therapy very early is that it blunts the peak viremia. So we can see that, sorry. So we can see that in early treated individuals, actually, the peak viremia was blunted by almost 3 logs when compared to untreated individuals. So we then wanted to investigate if early antiretroviral therapy suppresses the development of immune responses, or if early antiretroviral therapy helps to preserve the CD8 T cell responses. So, as I've mentioned previously, that most of our patients were identified HIV positive in FEBIC stage 1. We had some that were identified positive in FEBIC stage 3, and most of these individuals made responses. So, for those who were identified in FEBIC stage 1, some of them made responses and some of them did not make responses. So, this is an ongoing question in the field. If we want to know if these patients who are placed on immediate therapy, do they make HIV-specific CD8 T cell responses or not? So in this figure here, I am showing an example of an early treated individual that was placed on immediate therapy and had suppressed viremia. So we used tetramer staining to check for the HIV-specific CD8 T cells, and as you can see, we were able to detect HIV-specific CD8 T cells over time, despite, in spite of viral suppression. And here is another example of an early treated individual who suppressed viremia very early. However, this patient did not make any responses by tetramer staining. Also, the very same patient also did not have any proliferative responses by CFSC assay. So in this figure here, I am summarizing the CD8 T cell responses, HIV-specific CD8 T cell responses. So we used the 3 assays, tetramer staining, ELISpot assays, and CFSC proliferation assay. We used these 3 assays in combination because we wanted to increase the likelihood of detecting these HIV-specific CD8 T cell responses. In the interest of time, I didn't show the ELISpot assay result, but here it's just the summary of which patient made responses. So in blue, I'm showing the patient that had detectable HIV-specific CD8 T cell responses by 3 of the assays. In violet, I'm showing this patient who had HIV-specific CD8 T cell responses by either 2 of these assays. And in red, a small fraction here had detectable HIV-specific CD8 T cell responses by 1 of the 3 assays that we use. 34.8% of the patient did not have any HIV-specific CD8 T cell responses when using these 3 assays. So in summarizing this figure, we can see that about 65% of these individuals who are treated very early do make HIV-specific CD8 T cell responses. So the next thing that we wanted to do, we wanted to check the qualitative features of these CD8 T cell responses. In our group, we have previously shown that untreated acute HIV infection is associated with dysfunctional characteristics. So what we checked at CD127 expression, which is memory marker. And then what we found out is that in early treated individuals, HIV-specific CD8 T cells expressed higher CD127 when compared to untreated individuals, suggesting that these early treated patients may have CD8 T cells that may have potential for long-term survival. Having observed the phenotypic characteristics, we then wanted to check the functionality of these CD8 T cells. So we looked at tetramer staining and if the HIV-specific CD8 T cells secrete interferon gamma. So we stimulated the cells and then we stained with tetramer and then we gated on the tetramer positive CD8 T cells to check if these CD8 T cells do secrete interferon gamma. And what we observed is that the CD8 T cells from early treated individuals, most of the HIV-specific CD8 T cells secreted higher interferon gamma when compared to the CD8 T cells, HIV-specific CD8 T cells from untreated individuals. I'm also showing the summary figure of the 2 groups. We can see the significant difference in the secretion of interferon gamma between the 2 groups. So in conclusion, in our study, we were able to identify a hyperacute infection at the center of the epidemic, which is the nearest township. And then our results showed that a large proportion of individuals treated in phoebic stage 1 had detectable anti-HIV immune responses. Therefore, understanding of these immune responses in early treated people may be useful for future intervention studies. I would like to acknowledge my supervisor, Dr. Zaza Ndlovu, who's just right here, all the people listed in this slide, and most importantly, I would like to acknowledge the study participant and also the funders as well. Thank you.
Wonderful presentation. Thank you. Thank you. This is now open for discussion. Okay, I will start with a question. Have you been able to measure the CD8 response in tissues of any of the individuals at this time?
Yes, so we are looking at the lymph nodes at the moment, and what we found out is that some of the patients whom we did not detect CD8 T cell responses in peripheral blood, we were able to detect some responses in their lymph nodes.
That's wonderful.
Yes.
Yeah.
Hi, I'm Joe Huron from Chapel Hill. The 35% or so that didn't respond, did you look at the characteristics of the 2 groups, like age, maybe sexual exposure, how long they'd been sexually active, or any demographic characteristics that might distinguish?
So all our study participants are between the age of 18 to 23. And with their sexual behavior, I'm not sure, maybe our clinician can have that answered.
I just wondered about someone that might have multiple sexual partners, maybe exposures and HIV exposures previously versus a new single sexual partner or that sort of thing.
Okay, it's a good to look at that.
I'm Brigitte Autron from Paris. I'd like to know, on the same line, whether those individuals who are negative for HIV-specific CD8 T cells had much higher virus loads at time of diagnosis?
Yes, so most of them had higher. Some of them, although they're in phobic stage 1, some of them had higher viral load and some did not have higher viral load.
And had you an opportunity to look at those individuals later on and to check whether they accumulated memory, CD8 T cell memory? Or do you think the HIV-specific CD8 T cells is completely blunted in these individuals by early therapy?
Most probably, I would think that.
Did you have any chance to look, since you said you're looking in the CD8 cells in the lymph node, if these CD8 cells are able to localize inside the germinal center follicle? Because it's interesting in the context of TFH infection.
Yes, my colleagues are looking at that, and they've been trying to actually answer that question. They're still investigating that.
Yeah, we've looked in the lymph nodes and what is surprising with these CD8 T cells is that they can localize in the
Do I need to repeat the answer? Or? So he said that they look at the CD8 cells function in the lymph node, they localize in the germinal center, and you mean the HIV-specific CD8 cells, right?
So we haven't used tetramer in the lymph node to look at
Okay, okay. And you also said that they to produce low level of perforin and granzyme, cytotoxic molecules.
Okay, thank you very much for a really wonderful presentation.
Thank you.
Okay, next speaker is Kit Reeves. Kit is an assistant professor of medicine at the Harvard Medical School and has been conducted intense research on innate immunity in HIV and HIV infection. And the title of his talk is Memory-like NK cells exploit innate priming and alternative signaling mechanisms to enhance function and mobilize at HIV-SAV mucosal portals of entry. Thank you, Kit.
How do I? Oh, all right. So I am going to correct you briefly, Mirko. So I'm an associate professor now, because those titles are hard enough to earn that I think I'm going to.
Congratulations.
I'm going to give myself a shameless plug as opposed to citing a poster. So first off, I'd like to thank the organizers for giving me this opportunity to share some of our recent data on a recently described population of NK cells dubbed memory-like NK cells. And we're going to just really tease out some of the priming mechanisms and the signaling pathways necessary for these cells to function. And I'm going to talk about how these can interact in HIV and SIV infection, primarily focusing on SIV infection of rhesus macaques as a model. So first off, what are we talking about? What are memory-like NK cells, also referred to as Fc gamma receptor delta G NK cells? And as you can see on the left, this is exactly where the name comes from. This is the Fc receptor signaling complex. The Fc gamma receptor delta G memory-like NK cells lack the gamma signaling chain and coordinately lack the adapter SYK that goes along with the gamma signaling chain. Even though the name says memory-like, we'll come to that later, they do still require antibodies to grant antigen specificity, but they are more rapidly mobilized and proliferate upon secondary antigen exposure, which allows them some of these memory-like features. Now, in addition to that, they're important for us because they do have increased cytokine production compared to bulk NK cells, and they have much more potent ADCC functions alluding to this differences in the FC receptor signaling. Now, prior to our work, they had only been described in human blood, and one thing that I will point out that will become important in the talk as part of the innate priming mechanism is that they're massively expanded upon CMV infection. You can see that in the bottom right, is that in individuals that are seronegative for CMV, you have virtually no delta G memory-like NK cells, but following CMV infection, you have a high frequency of these cells in circulation. So since we are talking about SIV infection of rhesus macaques for a model, we just wanted to make sure that we could identify these cells in rhesus macaques. Very simplified gating strategy here. You see NKG2A positive NK, CD3 negative, and then you can identify the delta G NK cells. You'll see in the middle example flow panel, if I can get this to work, that they are negative for the gamma signaling chain, which is on the x-axis, but also lack expression of SYK. So we found that these were phenotypically analogous in humans and macaques. No clear differences in NK cell subpopulations based on CD56 and 16. But then you'll see in the bottom left corner, we had kind of a unique opportunity to explore this innate priming phenomenon with CMV in macaques because we can look in specific pathogen-free monkeys. And of course, you'll see here that in some specific pathogen-free monkeys, we have very few of the delta G and K cells. In normal CMV infected animals from a matched cohort, you see an increase in the delta G memory-like NK. And interestingly, in the final, you'll see in SIV-infected macaques, which are all CMV positive, you see even further expansion of these memory-like NK cells. And not surprisingly, in the graph next to that, the frequency of these cells correlates nicely with CMV serostatus. So of course, one of the benefits of looking in macaques is that we can ask questions about where these cells are localized within the body and what sort of functions they may be exerting there. So one of the first things we looked at just to sort of start getting at that was expression of lymph node homing molecules on peripheral blood memory-like NK. And you'll see that here is that the delta G memory-like NK express lower levels of lymph node homing molecules CCR7 and CD62L compared to bulk NK on the right. This suggests that these NK cells may not be circulating or surveying the lymph nodes as frequently as other NK cells. Now, in contrast, if we look at alpha-4 beta-7 as a surrogate marker for gut homing, we'll see that the circulating delta G memory-like NK cells express much higher levels of alpha-4 beta-7 compared to bulk NK, and that if you look in the bottom right graph right here, this is specifically looking just in SIV-infected animals. The alpha-4 beta-7 is even further increased in SIV infection, suggesting that these cells may preferentially home to the mucosa, which is consistent with other findings we've had on cytotoxic NK. So getting beyond phenotypic markers of trafficking and distribution, we wanted to see where delta G and K cells are distributed, and prior to the work we've done, this had never been shown because the lack of accessibility of some of these tissues in humans. So you can see on the right, these are just some example flow plots. Bone marrow, spleen, mesenteric and peripheral lymph nodes, liver, and colon. Basically, we can find these cells wherever we look, so they are systemically distributed. And then in a matched cohort of animals, we just look at the frequency. You find similar frequencies of them in PBMC and spleen, lower frequency at peripheral and mesenteric lymph node, slightly higher in colon, which would be consistent with the trafficking patterns that we've shown. And of course, much like what we saw in peripheral blood, in the spleen of SIV-infected animals, you see a massive increase of these cells, and then also in colon, you see a very significant increase of these cells as well, suggesting, much like the homing molecule data would indicate, these cells are expanding and homing to sites of potential virus replication, but are also expanding in general in response to infection. So of course the most important thing we want to ask about these cells is not necessarily where they are, but how they function and how they may be more beneficial in the context of mediating antibody-dependent functions. So just in the top right, or actually I'll start, in the top right, we thought, perhaps there's some differences in the binding of antibodies or antibody complexes to the delta G memory-like NK cells that are mediating this function, but as it turned out that is not correct. In this particular experiment, we took fluorescent HIV formed immune complexes, and we look at the binding to delta G memory-like NK cells and bulk NK, and we see no differences here. However, when you go to looking at the activation profile of these cells, this is just a simple CD107A assay in both humans and in rhesus macaques. Upon CD16 stimulation, you see much higher CD107A as a surrogate marker for cytotoxicity compared to bulk NK. And I'm not showing it today, but we also have consistent data for this in an actual killing assay. So that made us sort of question, really what's underlying some of the increased robust function in these cells. So one thing that we did find, you can see here, is that the CD16 density tends to be higher on the delta G and K cells compared to bulk and K, but more importantly than that is that the delta G memory-like and K don't have high levels of CD16 that would be associated with mediating ADCC-like functions until they've become exposed to CMV. And this gets back to the innate priming and the memory-like functions. Okay. So just beyond the functions themselves, we asked ourselves the question, what mechanisms may be responsible for mediating this enhanced potency, particularly specific to antibody-dependent functions? So one of the first things we looked at is how memory-like delta G and K may be transcriptionally different from bulk and K cells. I already mentioned that they lack the gamma signaling chain. They also have lower levels of SYK. And interestingly, you can see here in the flow plot as well in these graphs, they have much lower levels of EOMES and Helios. And this is an altered transcriptional profile that is usually, in NK cell biology, indicative of some different developmental program or potentially epigenetic modification, which is what we think is going on here. And it also fits in well with data from other memory and memory-like NK cell subsets, which tend to develop in the liver and also have this transcriptional profile. Okay. So beyond that, we asked ourselves, in terms of this function, we know that normally an NK cell, you can see here on the left, if they're going to mediate ADCC or some other antibody-dependent function, you have the immune complex binding here to CD16, and signaling is typically initiated through the gamma signaling chain and the adapter SYK, which you can see here if I can get this to work. But of course, these cells don't have that. And as I mentioned, they lack the gamma chain. They also have very low levels of SYK expression. And even if we do look at phospho-SYK and phosphoflow assay, you'll see that if SYK is present, it's generally inactive. So what that tells us is that this gamma chain SYK-dependent pathway of activation of NK cells is inactive in the delta G memory-like NK cells, but they still have much more functional potency. So with that, we started looking at alternate signaling pathways that the memory-like NK cells might be exploiting here. So this would not be surprising to anyone in this room. Of course, you see the gamma chain SYK pathway here. But also using CD16, NK cells can exploit CD3 zeta, which even though it is CD3, NK cells are known to express CD3 zeta, so don't confuse it with a T cell. And then they use the adapter molecules LCK and ZAP70. So we started wondering if they might be exploiting this alternative pathway as opposed to the gamma chain SYK. Now, as I said, most NK cells do express CD3 zeta. It's typical signaling for non-antibody-dependent killing, but in humans and primates, it can also be used for ADCC, and we hypothesized that this would be the alternate pathway. And so that's exactly what we see, is that in the delta G memory-like NK cells, you see much higher levels of CD3 zeta. It is phosphorylated at much higher levels in the delta G compared to regular NK cells, suggesting that it's an active pathway, and that the delta G memory-like NK cells also have higher levels of ZAP70, suggesting that the CD3 zeta ZAP70 pathway is being exploited by these cells to have much more functional potency. So just to summarize and conclude, as I presented, the memory-like delta G NK cells are more functionally potent than other Fc receptor-bearing NK cells, and they can be recalled much more rapidly after the functional priming that we see is necessary from CMV. They are systemic, and they mobilize in the mucosa during SIV infection, and then functional priming of the delta G NK cells depends on the prior CMV status. But interestingly, we have found that they use this alternate CD3 zeta ZAP70 signaling as opposed to gamma chain and SYK. And with that, I'll just close and acknowledge a few people in my lab, particularly Spandon Shah and Cordelia Manicum, who have done most of the work here, as well as collaborators, various institutions in our animal facility at Biomir, and then, of course, all the funders who supported this work. And with that, I'll close and take any questions.
Thank you, Kate. This talk is now open for questions.
Robbie Mallard from the University of Pittsburgh. Question I have is the priming of these NK cells, like with the T cell, you need a dendritic cell, signal 1, signal 2. Is there any factors that are important for driving this NK cell priming?
No, yeah, I mean that's kind of the golden question for this entire phenomenon is what exactly is specific to CMV that's necessary to prime them, and unfortunately I don't have an answer for you at this point. This is something that's been observed in mice and it's been shown in humans, but we don't really know what component of CMV is driving this. It's probably some sort of cytokine-based indirect effect, but that doesn't exclude other mechanisms.
Very good.
Yes.
Kate, since you're using the monkey model, there is any way to specifically deplete these cells and see how they impact on pathogenesis, viral control, and so on?
No, I mean, that's another good question, but unfortunately, the only thing that can really distinguish them clearly is this lack of the gamma signaling chain in sick. Surface expression of other molecules are different, but they're subtle. Like these differences in trafficking and homing molecules, they're very, very subtle. So at this point, we don't have a good approach to deplete them. The only thing that kind of distinguishes these cells, as well as other memory and memory-like NK, in both humans and in rhesus, is expression of NKG2C, which, due to difficulty with, there's no good cross-reactive agents for NKG2C in monkeys. So at the moment, we don't have an option.
Yes, nice talk. You have said that these cells express alpha-4-beta-7. Do you know what happens to these cells if you have looked at patients treated with vedolizumab, for example?
I have no idea. I think that's a fantastic question and something we'd love to look at, but we just haven't yet. Because it's really, I mean, this is something we've just kind of finished. And I think this was a new finding. This had not been shown in humans previously, so we just found it in monkeys. So I think the next step for us is to show that there's a similar expression pattern in monkeys, and then we'll, or in humans, sorry, and then we could maybe look in those cohorts. Great.
With patients with intestinal bowel disease, for example, independently of HIV.
No, absolutely. I think, I mean, yeah, it wouldn't necessarily have to be anything with HIV, but no, I think that'd be a fantastic idea.
Keith, do you think if you used a CMV vector, you could prime these cells?
It's a good question. So we're actually working on that. That'll be part of some stuff we're doing with Dan Baruch and Louis Picker vaccine.
That's a great idea.
Yeah, I think it will. That's kind of where this is coming from.
Okay.
Yeah.
Very quick question. What is happened to these cells when you put animal or human on antiretroviral therapy?
So that's a good question. So we haven't looked at that in monkeys yet, but I didn't show it today just because I was going to focus on the monkey stuff because the human work has been published. If you look at humans on antiretroviral therapy, you don't see a change in the frequency of these cells. So presumably, whatever additional expansion you get after HIV infection, it doesn't go down. It gets complicated, though, because I think, even in the cohorts, of humans we've looked in, the frequency of these cells is really driven more so by CMV than it is by HIV and SIV. And so I think, we have to do a little bit more due diligence because even though the serostatus, it nicely correlates, that doesn't tell you exactly how much virus is replicating. It's a surrogate indicator, but it's really driven by CMV more so than that. So presumably if they're getting immunocompromised, you get more CMV shedding, which expands these cells.
Thank you.
And if you treat for CMV with the activation?
We have not tried anything like that yet, but that's another good question. I think one of the next things for us is to actually take some of the SPFs, infect them with CMV, and then look at the development longitudinally.
Thank you for your lovely talk.
Thanks.
Thank you.